So Lee, thank you very much. Very inspiring lecture, and I'm sure there'll be a lot of questions from the audience. So what I would ask the audience to do is to please come forward, just introduce yourself quickly if you have a question for Dr. Hood. And we've left a generous amount of time for Q&A, so please just come on up and okay.
Lee, that was an inspirational and visionary talk. Thank you very much. I want to bring us down a level of science. So I was wondering, these IPS cells are really quite an exciting feature. But once you generate IPS cells and then differentiation back to another lineage, let's say neurons, does that wipe clean the epigenetic slate that might make a cell like a neuron become Alzheimer's-like or Parkinson's-like?
So we can't answer that question. I think that is a good question. I think many people feel that you do wipe the slate clean when you go through this process. But I don't think anybody's looked really carefully at that. And that's one of the things. We're actually looking at the differentiation of two classes of cells, neurons, which I talked about, and cardiomyocytes for cardiovascular disease. And with the cardiomyocytes, we can get such large numbers of cells. We're going to look throughout the differentiation process using all of the omics, including epigenomics, and just go right straight across and see how they compare with their normal counterparts. So we'll know that.
Maybe I'll just ask about some of these experts that's coming forward. So one of the major differentiating features between Seattle and between Vancouver is our single-payer system here for healthcare. And can you just speculate on how that's going to impact P4 medicine? How you think we may be at an advantage or disadvantage because of that?
Oh, I think you're at an enormous advantage, as I said earlier to some. My real dream is to have a nation adopt P4 medicine. And, you know, I've been thinking in terms of small nations like Luxembourg with 500,000 people, where, again, it's a single payer. It's a country that's run by 9 ministers, so you can really get things done. But the only countries we're looking at are going to be single payer countries. I think the heterogeneous system that the U.S. has would make it utterly impossible to do anything but a kind of a partial approach to this whole endeavor. So I think you're really in a good position to do something. Now you want to put framework for being able to do it. Yeah. That's it.
Hi, Dr. Hood. George Hoffman here. I'm the CEO of Network Immunology. So we're another systems biology based company. And you may be familiar with my father, Dr. Jeffrey Hoffman, who's working on a network theory of the immune system.
Mm-hmm.
And so it was incredibly interesting for me to be here. I really appreciate you coming up here, and it was incredibly interesting, generally the presentation, and very interesting how, especially how, I mean, 90% of what you're talking about is to do with networks. And from what I understand, from what I've heard, is it true that you were the founder or the discoverer of the IJ paradox of the network theory of the immune system?
My goodness, what a technical question for this.
Sorry, I apologize.
So I'm not a discoverer of the IJ paradox. What we did was disprove it. We showed that it couldn't be true. So, but we can talk about that. So, yes,
anyway, maybe we continue. I apologize. Maybe this is getting a little bit too technical for the general audience. But anyways, it's a pleasure to have you up here, and I really appreciate it. And perhaps to be continued at a later time. But just briefly, I mean, we've recently got some very interesting data discoveries in the area of a network theory of the immune system, and so it's very interesting to hear you talking so much about networks on various levels. And that's one network. The network theory of the immune system is one network that maybe hasn't had too much attention recently because of that IJ paradox, and we recently found some data to bring light back to that. So we're really excited to develop that and talk with you more about that.
Okay. Good luck with that.
Thanks so much.
Yeah. Mm-hmm.
Hi, I'm Robin Coop. I'm the instrumentation group leader at the Genome Sciences Center, so very much in the trenches on trying to make this stuff work in a sort of affordable way. And I think we're, with sequencing now, we're at a point where, getting genomics heavily involved in an active cancer patient case is within reach financially, because the cost of doing things like treatment, non-responsive treatment is very high. So that's you're already doing an expensive game at that point. But in terms of sort of public health, and particularly maybe think about the proteomics side, which I'm less knowledgeable about sort of current developments, how many orders of magnitude in cost are we away from, or factors of 2 or whatever in cost are we away from what we can do now to where you think this can really, this sort of longitudinal stuff can really work well?
You're talking about proteomics, are you?
Well, I think particularly, yeah.
Yeah.
I mean, maybe genomics. I mean, where do you think either of these are at?
Well, so let me talk about genomics. My view is that there is going to emerge a 3rd or 4th generation DNA sequencing device, which will be single molecule and electronic detection, great signal to noise. And my guess is it'll get the genome down to hundreds of dollars. And I think we'll get it down to $1,000 in 3 years and hundreds of dollars in 5 to 8 years. So I think it's going to go way down below what it is now. And I think those devices may well be able to identify the epigenetic marks, that is, the environmental modifications of the DNA as well. So I think that's going to be very exciting. And my guess is on the proteomics side of things, we can do proteomic runs for $200 now. So, I mean, assays of 100 or 150 proteins are well within reach of, regular clinical kinds of assays. Now, if you did that twice a year, would people be willing to pay $300 to get, I mean, we're not going to have the 2,500 for a while, but we can look at the major ones. We'll have all the major organs very soon. I think they will. One of the places we're thinking about starting with some of the P4 medicine and wellness factors is in executive health plans, where people that are really well off don't mind paying extra money to get an enormous increase in the amount of information. That may well be the driver that puts the wedge into getting some of these things started, even when they're not at the microfluidic protein chip, far less expensive, far more measurement stage. But with hundreds, you could do a lot still. If you had your heart and your liver and your brain, and any other 2 or 3 organs that you selected, checked on a regular basis, that would catch an awful lot of disease, obviously.
I find those things intriguing, certainly with the sequencing. As the sequencing gets cheaper, the analysis continues to be daunting. And the way we're doing it now, sample management is daunting and so forth.
But I think the analysis is really going to get to be very easy as we figure out how to do it better. I mean, the problem now is we're just putting together all of the tools, but there are companies out there now that'll take a human genome sequence and do the clinical analysis, and they can do a really great analysis for $1,000. So that's got to come down, but it's a start. It's a start.
Thanks.
Jim Russell, Vancouver. I'm a professor at UBC and a co-founder of a little company, Serious Genomics. So I'm very interested in this personalized medicine approach. And one of the things that struck me, I'm mostly a clinician trial scientist, and as you probably well know, many drugs that were discovered, insulin as an example, have turned out to be far more complex than we ever imagined. They don't just control blood sugar, they interact with a variety of signaling systems in different cells, different tissues. And then there are many drugs that have failed in the conventional paradigm that you talked about that's been 50 years. And so I wonder, using a systems biology approach, if you could somehow take advantage of the chemistry of failed drugs and an evolving understanding of the biology as you defined it from the DNA, say, to the whole organ, And could you resurrect dead drugs and reformat them more carefully to a narrow disease or a narrower subtype of a disease based on a molecular phenotype? Do you think that's a complementary or a better, or compared to discovering new molecules and new receptors, almost defining new diseases, which is kind of where big pharma seems to be going? And it seems to me maybe your approach could take advantage of some dead drugs and reformat them and actually find disease or subtypes for which they could be very beneficial.
No, I agree with you entirely. In fact, one of the exciting ideas is all the drug companies can pull forth all of their drugs that passed the safety tests but didn't pass the efficacious test. And then with all of these different stratified patient populations, you could go through and begin testing those on the individual. There is one enormous complication in this, and that is my deep conviction in the end is that most diseases will be treated not by one drug, but they'll be treated by 2 or 3 or more. And that means you have to add to what we're talking about a combinatorics that really gets out of control. So what we have to do, if we're going to do that, is we have to get really good cell-based assays that reflect the patients. And I really think we can do that with some of these strategies that we've talked about and so forth.
Well, that was almost the question I had. Taking your neurons as a surrogate, each individual human would have a unique neuron that would have a unique database of DNA and perhaps an environment at that age, at that stage in their life. We've actually designed these middle trials, but screening devices, screening assays to then take that list down to, because the big thing, as you all know, is the very great expense in terms of time, money, and humans for the clinic.
And the other thing is, once you've got your genomes done, we're going to learn more and more about modifier genes. So we're going to be able to start classifying patients into distinct groups based on the modifying genes and how they respond to drugs. It's pharmacogenomics in a positive rather than a negative sense, basically.
Thank you.
Okay.
Hello, I'm Anna, and I'm an informatician at a small startup company here. I have a question that's a two-parter, but very quick. The first one is, how easy do you think it's going to be for physicians to part with the current paradigm that they're operating in? And what does that mean for people getting medical training now? The second part is, if we move towards an approach that's really driven by patients, I mean, scientific literacy isn't great in the general population. Even if we educate them, that might be biased from different sources. So how vulnerable does it leave the patients if, say, physicians don't support it, or it's very heavily patient-driven?
Well, look, I think physicians are going to have a hard time accepting anything new. And my conviction is most rational people, if you can show them how they can really do things better, will, if grudgingly, begin to accept it. And that's really the purpose of some of these pilot projects that we've really set up. The education of patients, frankly, I think in the end, patients are going to do an awful lot of educating themselves. I mean, you go to companies like 23andMe that have done these genetic analyses on the genome, and you see patient populations that are unbelievably enthusiastic and committed. They've identified something like 6,000 or 7,000 patients with Parkinson's disease who have their families enthusiastic about being willing to participate. And they're really driving this thing forward in very powerful ways. And I think the analogies, which is really a good one, is again the AIDS analogy. It was the AIDS activists that really drove the acceptance of triple drug therapy that converted AIDS from a fatal disease to a chronic disease. And I think patients are going to do exactly the same thing with the new medicine. And I think they're really going to push physicians. So it's what it will be to be a physician is really going to be an interesting thing to talk about, think about. Yeah. And how many kinds of physicians are we going to have?